For this lab, we are going to be looking at disinfectants and antiseptics. This is such a cool lab to do in the actual lab classroom, because what we end up doing is having you guys bring in all different kinds of disinfectants and antiseptics. Things that I encourage you to bring in are things that you use in the home, something that you use to clean a surface or part of the body that you would expect to be a disinfectant or to be an antiseptic. And so I tend to have students bring in some cool samples, anything from Windex to 409. I've had students bring in the makeup brush cleaner or some other personal cleaners. I'll just leave that one there. But we get to see all of these different results of these products. So unfortunately today we're going to be doing it online, but that's okay. We'll walk you through it and I'll show you some results from classes of past. So in this lab, we typically have you guys break up into groups of 4 to do this, looking at different types of antiseptics and disinfectants. So let's just kind of walk you through overall what we're going to be doing. For the disinfectants, we end up with nutrient agar plates with three plates with each having its own culture on it. So we'll have Staph, E. coli, and Bacillus megaterium. The plates get prepared with a lawn of bacteria, and then we have these discs, and they essentially look like little hole punches, and these are filter discs that soak up the liquid of whatever you're trying to test, and then we aseptically transfer these little discs onto the surface of the plate, onto the culture, and then we incubate it and see how well that disinfectant or antiseptic performed. We will worry about the antiseptic plate, the antibiotic plate in a future lecture, so don't worry about that now. Let's move on to the actual methods. So we have you work in groups of 3 or 4, and you're going to take a nutrient agar plate and essentially create a lawn of bacteria. So if this is our plate, you're going to take a swab and coat the surface of that plate back and forth and back and forth and back and forth and back and forth and back and forth. So that way all surface, I call it, you're chasing the circle, or you're chasing the corners in a circle. So continue until you find all the corners in a circle, and then you should be good. Each plate, you're going to have 3 plates, 1, 2, and 3, is going to have B. meg on it, Staph aureus, or E. coli. And so you're looking for how those particular species performed against the different types of disinfectants or antiseptics. Now, why did we choose each one of these species? Well, E. coli, what type of a gram reaction does E. coli have? What type of a gram reaction does Staph aureus have? And then Bacillus, why might that be important? And we'll discuss that as we go over the results today. So we end up placing the disinfectants and antiseptics in what we call a depression plate. And it looks like a miniature egg carton. So if this is that, you'll have little wells, and those wells can hold liquid. And so you put disinfectant number 1 in here, and then you'll put antiseptic number 2 and 3 and 4 in our little well plates. Then what you end up doing is taking aseptically, you end up taking one of the little filter discs out, soaking it in that disinfectant antiseptic, allowing it to absorb some of that liquid, and then you very carefully place it on a plate, and then let the plate soak in and the disc adhere to the plate, and then you flip it and put it in the incubator. So I'm going to jump back to this slide because here are our filtered discs. So in this experiment or this setup, we've tested a plate of E. coli. So E. coli is covering the surface, and then this filtered disc has been dipped in Listerine mouthwash. This has been dipped in Lysol, tea tree, I would assume tea tree oil, and this one has been dipped in bleach. And we incubate at 37 degrees Celsius, and then we let that sit, and we're picking up with day 2. So at this point in time, we have incubated our plates, and we're going to read out the results. So we are testing different disinfectants and antiseptics. Let's define these terms. So a disinfectant is a chemical that kills or reduces microbial populations on inanimate objects. So where you're going to be seeing disinfectants being used, let's say in the home, is perhaps to lower the microbial load of your cutting board, of your sink. Perhaps you'll use disinfectants when cleaning the bathroom or door handles. So disinfectants are going to be used in the home. We'll see them used in the medical industry all the time to disinfect surfaces. An antiseptic, however, is something that's going to be used on living tissue. So something like a betadine or hand sanitizer, it would fall into the category of an antiseptic. An important thing to note is that we can have disinfectants that can also be antiseptics. And so a lot of our alcohol products, ethyl alcohol or isopropyl alcohol, can be both a disinfectant as well as an antiseptic. So what we're going to do with our results is we're going to measure the clearing on our plates, and that clearing is called the zone of inhibition. And that zone basically means that there was likely death of cells within that zone, but we'll kind of talk about why maybe not all the cells are dead in that zone in a future lecture. So we're going to measure the clearing of the disc, and in disinfectants, antiseptics, it's really important to measure from the edge of the disc to the edge of the clearing. So that's our little zone of inhibition there. In our antibiotics, we're going to do a different type of measuring, but this is from the edge of the disc out. If you have more than a 2 millimeter clearing, that organism is sensitive to that particular chemical. If it is less than 2 millimeters, it is resistant. Now, if you have a metric ruler, I encourage you to take a look at how small a millimeter is. So when we say 2 millimeters, it really is a very, very small clearing. So whether or not you get a large clearing or a very small clearing, if it is over 2 millimeters, it's still considered sensitive, meaning it's effective against that particular organism. So this is a picture of Bacillus meg, and we've created a lawn. They've tested 4 different chemicals. Our discs have been dipped in those chemicals and then placed on the plate. That plate has been incubated for 48 hours, and it looks like if between Listerine, honey, ammonia, and pine sol looks to be the only chemical that B-Meg is sensitive to. So we've provided for you results of plates. This is E. coli. Now I asked you to think about E. coli and what makes it unique. E. coli is a gram-negative cell. So think of that cell wall as the outer membrane, the very thin peptidoglycan layer, and then our inner membrane. Staph aureus is gram-positive. So it has a very thick peptidoglycan layer, but it does not have an outer protective membrane. B-Meg is a gram-positive cell, But what's interesting about B. meg is it produces endospores. So yes, the vegetative cell might be sensitive, but what about endospores? So we have inoculated these plates with each type of bacteria. We have taken a disc and dipped it in the various disinfectants and antiseptics listed below. And then we've placed that disc on the plate, incubated it, and we're now going to look at the relative clearings of these. So you can see in the top right corner, we have number 1. So that was the disc that was treated with hydrogen peroxide, white vinegar, bathroom cleaner, and then mouthwash. Now the bathroom cleaner isn't defined more than just bathroom cleaner, so let's assume something like a Lysol or a Clorox or like a scrubbing bubbles kind of thing. So as we look at this, you're looking to see, 1, is there a clearing, and then 2, how large is that clearing? With the measurements for disinfectants, antiseptics, again, you're going to take it from the edge of the disc to the edge of the clearing. So you can see that there is a clearing here. Let's assume for all intents and purposes, this is greater than 2 millimeters. So hydrogen peroxide for B. meg, Staph aureus, and E. coli is an effective antiseptic for those particular species. Now I want to focus in on E. coli. Look at E. coli here for mouthwash, number 4. I barely see, if not at all, a clearing right there. I'm going to say that that is resistant to mouthwash, which is kind of interesting because mouthwashes and toothbrushes and things of the dental sort are kept in your bathroom. And E. coli is frequently deposited into toilets in the bathroom. I have run into people that have stored their toothbrushes in mouthwash upside down to, disinfect their toothbrush in between uses, or they just store it in mouthwash. They've done studies where they can look for basically fecal bacteria, fecal coliforms, in a bathroom. So when a toilet flushes, how far does E. coli travel? And they've tested it and found that E. coli was present on a number of different surfaces inside the bathroom. And so if you're storing your toothbrush in E. coli, if you're storing your toothbrush in mouthwash, that mouthwash really isn't effective against E. coli. And if you're in a bathroom that your toothbrush is out on the counter next to a toilet that flushes a lot of times, you're not disinfecting your mouth or your toothbrush, which is kind of an interesting thing. So this is a chart that is in your notes, and I know it looks like a mishmash of colors and symbols and chemical names. So let's break this down and I'll kind of explain to you what's happening here. So we have taken our results from our plates and we have measured that 2 millimeter zone of inhibition. And if it is less than 2 millimeters, it's resistant, or R. And if it's greater than 2 millimeters, it is sensitive. So these are color-coded based upon chemicals, and there might be multiple groups that tested the same chemical. So for instance, I'm going to start up with the alcohol group up here and B. meg in the alcohol group. So a group tested Listerine with B. meg, Staph aureus, and E. coli. So that's that mouthwash. And for all 3 types of organisms, it was resistant. We had 2 groups that tested rubbing alcohol in black. So one group got resistant against B. meg, resistant Staph aureus, resistant and sensitive for E. coli. So maybe that was a alcohol that was newer, not sitting on the shelf for very long, or perhaps that was discrepancy with measuring. I don't know. Hand sanitizer, we had 60% alcohol, BMEG sensitive, resistant, resistant, sensitive, resistant, sensitive for staph, and sensitive, sensitive, resistant for E. coli. So there are a number of different brands of hand sanitizer, and some hand sanitizers has isopropanol. I have a hand sanitizer that has ethyl alcohol in it. So some different varying results there. Scope is another mouthwash. This is just a generic mouthwash. And then we also tested a 90% rubbing alcohol. So you can kind of see that there's variability within the chemicals themselves and even with the groups that tested particular chemicals. When we get down into our quaternary compounds, these disrupt cell membranes, and we've got a number of different types of chemicals that fit in the quats. They are going to target mostly bacteria, but we do have some fungi and virus that can be targeted by our quaternary compounds. So we had a number of different groups test a Lysol compound. And in my classes, I've had students test Lysol wipes or aerosol Lysol or the spray Lysol, and then sometimes it's a toilet bowl cleaner Lysol. So I don't know with Lysol what these groups tested. And there is, again, some variability among the groups. We have red for our BMEG, so mostly sensitive, one resistant group there, mostly sensitive for staph, mostly sensitive for E. coli. And if you read the side of a Lysol bottle, it is advertised as a disinfectant antiseptic, and it is advertised to target organisms such as E. coli and Staph aureus specifically. Another type of mouthwash, we have Bactine, Disinfectant Spray 409, Pine Sol Simple Green. So you can go and look at the chart and see that there was some variability. Some are resistant, some are variable. Halogens. Halogens are that column on the periodic table. They have action as free radicals within the cell. They can target cell membranes. They can target proteins. We're going to see a lot of different types of targets within the cell. They are oxidizing agents. So oxidation is loss of electrons. So they're going to steal electrons away from important enzymes inside the cell. So bleach, I found across the board, bleach is a fantastic disinfectant. It tends to be sensitive to all three species that we have. In the industry, I worked as a veterinary technician for 3 years in emergency vet med, and anytime somebody came in with an infectious case, we would always recommend that the owner disinfect the surfaces with a 1 in 10 bleach solution because it was just an effective compound. Lysol toilet bowl cleaner, you can see it's sensitive across the board. Me personally, I would care mostly that Lysol toilet bowl cleaner is going to be effective against E. coli because, again, that's what you're depositing inside of the toilet mostly. So is it active against E. coli? Clorox wipes, iodine. Interesting, this results for iodine, we had sensitive resistant, and sensitive sensitive. I tend to see iodine in my class results where we have a sensitive across the board. Iodine is something that is going to have a very particular shelf life because it is a halogen. It's stored in those little amber bottles, so it does go bad. It has a very short shelf life, so we want to pay attention, and perhaps we had an iodine that was a little bit older than what we were looking for. If we go down to our peroxygens, those are free radicals. Hydrogen peroxide is a fantastic antiseptic. Using it on tissues, it bubbles up, it causes that reaction, but it also does target some of these cells as well. Clorox green wipes, we have Clorox healthcare wipes, hydrogen peroxide, 3% hydrogen peroxide. And then none of this particular set of class results did not test any of the others. I'm going to pop up one we have some other cleaners on here, as well as our biguanides. So I'm going to talk about the ones we haven't talked about. So biguanides, we have targeting against the cell membrane. A commonly used biguanide in the medical industry is chlorhexidine. You'll see this in scrubs or soaps or hand washes in the hospital or medical setting. It tends to be sensitive across the board. These results have a resistant. In my class results, I've seen sensitive across the board. Toilet bowl cleaner, let's hop down to the others. Vinegar and acetic acid is used in the organic industry as an alternative disinfectant antiseptic for treatment of food products, and to get that label organic, you can use organic acetic acid. Now, vinegar is, I believe, 3% acetic acid, so it's a little bit lower content acetic acid than what they use in the industry. I tend to suggest using vinegar if you're opposed to bleach, but just understand vinegar isn't as effective against certain types of organisms. And in fact, this chart here says it was resistant across the board. I've seen it where it's resistant in Bacillus meg, but sensitive in Staph and E. coli. And again, that might be the type of vinegar, apple cider versus the white vinegar, or acetic acid that has a higher percentage, like maybe a 5% acetic acid. Pine Glow, I'm going to assume that is a floor cleaner, or something along those lines, something with some orange in it, sensitive across the board. I did a lot of work in grad school with essential oils and extracts. There are essential oils and extracts that are very effective against some pathogens. I worked with oregano essential oil, and it was very useful against species like Salmonella and E. coli. So there are alternatives out there if you're not a bleach person. And in this day and age where we are disinfecting almost every surface of the house, I'm going to remind you it's very important to read the side of the bottle and read the label, because every disinfectant that claims it's a disinfectant has been tested, and there are time limits and exposure times and things that need to be adhered to in order to get proper cleaning. So I'm just going to reinforce that. Read the side of the bottle. If you're using Clorox wipes, it is effective against these bacterial species, but only when you maintain that surface being wet for 4 minutes. So that is a product that you're going to have to continuously keep that surface wet in order for it to work, which may not be how you actually use that actual product in the home.